next speaker, Dr. Angel Peterchev from Duke. Angel.
Good morning, and thank you for the invitation. I'm going to provide what I formulated is 6 pieces of advice to researchers venturing into animal studies of transcranial electrical and magnetic stimulation. Next slide, please. These are my disclosures and funding for the work that was presented. Next slide. So to reiterate something that was brought up already by a couple of previous speakers, it's important to keep in mind what are we doing when we're delivering transcranial magnetic or electric stimulation. And in both cases, we are applying external stimulation parameters. Next click. That can be defined as the dose of stimulation to deliver what I call an electric field. Dr. Lisenby delivered the received dose in the brain, and in both electric and magnetic stimulation, it is an electric field that is induced in the brain. And it is something important to keep in mind as we consider different modalities like TES and TMS for delivering stimulation to the brain. Understanding this active ingredient or this received dose can then inform the parameters that we select. And it's very important to keep these relationships in mind as we go across different species. Next slide. I'm also showing this study by Dr. Opitz's lab and Dr. Alexeychuk, which is really elegant demonstration of why we have to consider dosing very carefully when we work with different species. Basically, this slide shows us that if we're doing transcranial electrical stimulation with the same current in a mouse versus a human, we get 100 times stronger stimulation in the brain, 100 times stronger electric field in the brain in the mouse than the human. And for magnetic stimulation, the opposite is true, basically. In the mouse, the electric field will be only about half of that in the human. So for the same external parameters, we get a lot stronger stimulation with TS in mouse and a lot weaker stimulation in mouse than in the human. That's why it's really important when we talk about translatability to characterize what is actually delivered to the brain and do that properly. Next slide. So the third advice, therefore, is measure the delivered electric field whenever possible. So there are different ways to do this, and that really depends on your experimental setup. I'm illustrating this here with one example of a collaboration here with Duke, with Dr. Turner, Dr. Dennis Turner, who's measuring the effect of transcranial electrical stimulation on blood flow in mouse. And that was measured with laser Doppler. In addition to the laser Doppler that could detect the blood flow, he had an electrode, a glass electrode that was measuring the basically electrophysiological response in the brain. And initially he had one electrode, and he had these nice plots of blood flow versus current delivered to the external electrodes, and then also the electrophysiology basically with one channel in the brain. And I said, great, but we don't know actually what is the electric field delivered in the brain, and this is critical to interpreting this. So I said, why don't you add another electrode, and then you can take the difference in voltage when you apply the current, and you can actually estimate the electric field just by difference in the voltage and dividing by the distance between the electrodes. So what we got in the end is on the next slide. Basically, there were several mice, 8 mice were run without this 2 electrode measurement to get the electric field, but then the last 3 were done with 2 electrodes. And based on this, we could create a relationship between the applied current and the electric field delivered to the brain. So then we used either that measured electric field in these 3 mice or the estimated electric field for the other 8 mice based on their current and this relationship to have a plot that to me was the crucial element of dosing here. What is the relationship between the intracranial electric field and the cerebral blood flow change? And indeed there was a positive correlation, right? There's some limitations. We estimated the field for some animals versus measurements, and so on. The important point, though, is that this really casts very important insights. When you look at the x-axis, it starts from a couple of volts per meter or millivolts per millimeter, which is above what can be delivered tolerably in humans, right? Which is typically below 1 volt per meter without anesthesia. So this is an important observation that these phenomena are very interesting, but you can't deliver them with conventional subthreshold TES. You have to do something else, right? And that came from measuring the electric field that's delivered and associating that with the response. Next slide, please. Okay, so a measurement like this takes basically 2 points and estimates the electric field by taking the difference, right? So the electric field is really a vector, right? It's more complex than that. And that's where electric field models are really helpful in understanding the full distribution of the electric field with all of its details. So in some situations, you may be able to just make an electric field model and not have invasive recordings. In some cases, you have the privilege, like in the study that Dr. Frawley just talked about, to have both recordings and also construct detailed electric field models to understand the field distribution. So I'm showing here how one goes about that. It's important to have the right kind of imaging. For very small animals, you may want not only MRI, but also X-ray, CT imaging to reconstruct properly the skull. And when you work carefully, you can really create a nice model that incorporates the biology of the animal, as well as any implants that are relevant to modifying the electrical flow. And this is a ferret receiving transcranial electrical stimulation. So you can see some of the results on the next slide. So here you can see these beautiful models of the electric field distribution within the brain, and in black boxes we have the regions that are of interest in this particular study. So here we had the privilege of having electrodes implanted in the brain, so we can measure the potentials produced by the transcranial electrical stimulation, and then we could compare the model with the measurements, and they fit very well, as you can see, in terms of induced potentials for different current levels. So you see basically the diagonal line, and the dots in red, green, and blue are that the measurements give you localized information about the electric field, in particular clusters where the electrodes are, and then the simulation, if it's validated or even calibrated by these measurements, give you the full distribution across the brain, and you can do further quantification based on that. So it's a very nice marriage between measurement and modeling here. Next slide, please. And now, how can these models be useful? So here's a study where we just did the electric field modeling because we did not have invasive recordings. In this study of non-human primates receiving transcranial electrical stimulation. Also, there was transcranial magnetic stimulation in another arm of this study. So the question that we had, you can see that we constructed detailed electric field models, and that paid off. Even though we just had 4 subjects, it really provided insight into individual variability that we see. So this is clear on the next slide. So in these non-human primates, we measured a couple of responses. The multi-threshold, that is the threshold at which you get the finger twitch. And we compared that, so the experimental results are on the y-axis. On the x-axis are the predictions of the model in terms of basically the strength of the electric field that you get in the brain for given transcranial electrical current applied. So it tells you, for the same current applied, how strong is the field in the brain? It's this kind of coupling coefficient, right? And you expect that if you have stronger electric field in the brain for a given current, you have a lower threshold for activation, assuming that the threshold in the brain is about the same, and therefore you have different current that is applied. And you can see that this is indeed the case with these correlations between the motor threshold and also the seizure threshold. We're studying electroconvulsive therapy, and we wanted to see whether it makes sense to individualize the amplitudes to induce a seizure. And the answer was that yes, that seems appropriate, and it is explained in large part by the delivery of the electric field to the brain. So why is this important? One reason this is important is that in conventional electroconvulsive therapy in humans, they use fixed current. And here we show that, well, actually, it may make sense to individualize this current, and there's an explanation for it. They feel that is delivered to the brain on an individual basis. Next slide, please. So this is my last piece of advice. And it is for those of you who want to study TMS in small animals, please consider doing that with non-magnetic means. That is, consider basically using electrical stimulation instead. And the reason is the plot on the left, where you see that if you try to build a small enough coil to really reproduce the focality and strength of TMS, of what is delivered to the brain in terms of electric field between human and a mouse or a rat, it's basically impossible to do. You end up with a small coil that would just evaporate if you apply the current that's sufficient to really produce focal stimulation. So what can we do about that, right? We can't build such small coils, but we can reproduce the focality of stimulation with a pair of electrodes. And in this modeling study, we studied whether you can put them epicranially or through the skull or on the cortical surface. And you get the results on the next slide, which basically tell us that, yes, this is a good approach. You can use a pair of electrodes. And one more click, please. On the left axis, you see the focality of stimulation. And on the right axis, you see the current that is injected in the electrode. So basically, you can reproduce the focality of a conventional figure of 8 coil in TMS with stimulation that is with electrodes, maybe epidural, kind of going through the skull. And you can use only 0.5 milliamp of current to reproduce what will take 3,000 amps of current in a human TMS coil. So keep that in mind next time you try to do TMS in small animals. So that's all. And in the next slide, I acknowledge my collaborators in these studies, and also I summarize my piece of advice, which I hope will be helpful to you. Thank you.